myself Dr. Vivek Kumar, Professor, Himalayan School of Biosciences, Swami Rama Himalayan University, Jolly Grant, Dehradun. I am again with you with a new topic on endophytes. That is the endophytes' indirect benefits to the host plant. As we discussed before, that endophytes are the microorganisms which reside inside the plants. Once they reside inside the plants, whether it could be bacteria, fungi or actinomycete, they benefit. Generally we assume that, and it has been observed, proved in experiments that endophytes within the plants, they impart certain benefits to the plant. And there are certain benefits, they could be direct or indirect. So today we will discuss some indirect benefits. So we can say that endophytes are fungal and bacterial organisms that inhabit the plant endosphere without harming their host or without any apparent observable harm to the host. So they live asymptomatically in the plant cellular environment and they perform symbiosis specific functions. Symbiosis specific function means they provide something to the plant and in return they get something from the plant. So such as synthesis of secondary metabolites, these are provided to the host by the endophytes, or signaling molecules that function as internal and external stimuli during the mutualistic interaction of endophytes and the host plants. And the endophytes, they also produce biologically active metabolites such as immune responsive compounds, plant growth promoters, antimicrobial volatile compound, insecticides, antioxidants and antibiotics. So all these secondary metabolites produced by the endophytes, they help their host plant. Now, since abiotic stress also include one of the different stresses, non-living stresses. So we discuss the stress tolerance. So when the plants, they are exposed to diverse stresses and which are the result of antagonistic or toxic substance in the environment. These could be anything: higher temperature, low temperature, high salinity, low salinity, high pH, low pH, excessive concentration of toxic metal ions, and other things are there. So these could be nutrients or essential iron limitations, and there could be some biotic stress inducers also. So both biotic and abiotic stress, they contribute about 30 to 50% of the agricultural loss all over the globe. So that, as we have discussed before, the temperature, the salinity, the drought, the trace metals, even the flooding also, the nutrient deficiency, they all are considered abiotic stress. Abiotic means non-living stress, biotic stress means the living stress. Biotic stress we will discuss in another lecture. So the biotic stress, they are we discuss something, they are induced by pathogenic microorganisms. It could be insects, microbes, nematodes, maybe some herbivores also. So therefore the future studies aim to develop eco-friendly technologies which will increase plant resistance to both abiotic as well as the biotic stresses. So a promising strategy could be the generation of stronger and healthier plants with an improved immune system. With the improved immune system means that the plant, they are going to protect themselves. So endophytes have long been known to fulfill these criteria and they provide excellent systems for agricultural application. Agricultural application means they can be employed as potential bioinoculants or biofertilizers. So the microbial strategies range from immediate activation of responsive systems which directly and specifically they counteract the stress, and the production of anti-stress metabolites that strengthen the plant. So the anti-stress metabolites, they are produced by the associated endophytic bacteria of the host plant. Now this picture shows the different stresses which influence the plant growth. This could be abiotic stress, it could be the biotic stress. The abiotic stress here we can discuss before: drought, cold, temperature, flooding, salinity, trace element and nutrient deficiency. Biotic we will discuss in the next lecture. That could be by microorganisms or could be by some living organisms, something like that. So when there is a abiotic stress, so there is, we can say, there are side effects on the plants like decrease carbon dioxide fixation, enhanced rate of transpiration, leaf aging, accumulation of reactive oxygen species, reduced nitrogen fixation, disrupt hormonal production. So, all these features, they adversely affect the health of the host plant. So, once the host plant is associated with the beneficial endophytes, then these harmful aspects of abiotic stress, they are mitigated. So, endophytic vectors support plants to combat drought stress using the production of volatile compounds, abscisic acid, ACC deaminase enzyme and indolacetic acid. So, moreover, the enhanced antioxidant activity and osmotic adjustment participate in the endophytic effects also. The salinity tolerant Staphylococcus have been reported to improve the growth and salt tolerance of Saliconia species plant either individually or in combination with the other microorganisms. So, using this Staphylococcus plant, so it has given salt tolerance or resistance to the host plant. So, the endophytes also manage host resistance to pathogens. They also includes niche competition. So, this is done by production of defensive metabolites such as antibiotics, certain antimicrobial compounds and structural compounds as well as the induction of immunity or systemic resistance in the host plants. So, the plants experiencing biotic as well as the abiotic stress, they produce elevated concentrations of ethylene which are synthesized from ACC. So, the production of defense hormone ethylene, it restricts growth and hence affect the development of plant journal, if we generally discuss this aspect. So, the improved growth and stress tolerance of 2 wheat varieties in one experiment inoculated with ACC producing bacterial endophytes. So, it was observed that an advantage of ethylene producing endophytes may be the local synthesis of hormone in the symbiotic tissues, which might ensure that the ethylene induced defense occurs only in tissues when they are exposed to the stress. So, we can say during stress ethylene is produced. Now, there are certain indirect and direct benefits of the bacterial endophytes on the plant growth. Indirect benefits means, today what we are discussing, they suppress the host plant disease by production of ammonia, by production of antibiotics, certain volatile organic compounds. They induce the systemic resistance in the host plant and they protect the plants from the pest and the herbivores, and also the endophytes, they play a significant role in phytoremediation. So, they can remove or we can remediate the metals or the hydrocarbon contaminants. So, endophytes produce ACC deaminase, they can alleviate the impact of elevated ethylene concentrations on stressful plants through the hydrolysis of ACC into alpha ketobutyrate and ammonia. So, ACC has been broken down into ammonia and alpha ketobutyrate. So, the stress plant so they can utilize ammonia as a source of nitrogen and the energy liberated from ACC decomposition over the growth. So, the multiple combinations of beneficial features of endophytes such as combinations of ethylene, siderophore excretion, exopolysaccharide production, they are important aspects to strengthen the plants under stress or nutrient limitations. Now, if we discuss the endophyte based phytoremediation. So, phytoremediation is a promising tool for cleaning soil, water, air, and endophytes assisted phytoremediation is used for metal bioremediation in the soil to allow or promote plant growth in previously contaminated soil. So, people have observed the effect of Enterobacter, Sphingomonas, Methylobacterium on the growth of zucchini plants on DDE contaminated fields. So, plants inoculated with 3 strains as we discussed before, or singly or in combination, they have shown enhanced weight of the host plant. The sweet white clover plants, another experiment, planted in soil contaminated with diesel up to 20 milligram per liter. So, when the plant was inoculated with the hydrocarbon degrading endophytes, for example, your Pentoa, Stenotrophomonas, Pseudomonas and Flavorbacterium, they showed increased biomass and successfully overcome the growth inhibition observed on the non-inoculated plant. So, we are discussing this experiment with reference to the diesel which was present in the soil up to 20 milligram per kilogram. So, the endophytic Bacillus sapiens, another experiment, exhibited multifractional properties including efficient degradation of C12 to C32 alkanes of diesel oil and polycyclic aromatic hydrocarbons under hypersaline conditions. So, there are 2 stress salt as well as hydrocarbon. So bacteria also produce biosurfactants, which resulted in stronger growth and biomass production in the inoculated plants as compared to the uninoculated or the controlled plants. Now with reference to disease control, so agrochemicals generally they are considered the main method for controlling the disease. However, many of the applied substances they have toxic effect on the soil, on the plant. When we consume the plants, obviously indirectly we are also consuming those pesticides. So the application of endophytic bacteria is a we can say another choice. So it could be an environmental friendly choice. They are farmers friendly, eco friendly and pocket friendly. So when we use potential endophytes, they can be used to eliminate or control the growth of phytopathogens. So they can restrict pathogen invasion into plants by direct and indirect mechanisms. The direct mechanism describes a competition between the endophytes and the phytopathogens, in which the endophytes they restrict pathogen growth through a secretion of inhibitory metabolites. In indirect mechanism, what happens? The endophytes stimulate the plant's immune system or increase plant resistance towards the phytopathogens, or we can say the upregulation of the defensive genes of the host plant. So the endophytic species of Pseudomonas aeruginosa, they produce 2,4-diacyl fluoroglucinol and other carboxylic acid, pyrrolene nitrine, hydrogen cyanide, which suppress these chemicals, they suppress the growth of the phytopathogenic fungi. And the most common endophytic bacterial species used in phytopathogen control the Bacillus species, since they produce lipopeptides which hydrolyze fungal hyphal membranes. So destabilization of fungal membrane promotes nutrient leakage from the fungal membrane, hence so the virulence of the fungi is reduced. Then there is the competition for space and nutrients also. The niche competition means the competition of endophytes with the deleterious pathogens for space and substances. The competitive interaction was observed between endophytes and the aggressive pathogen Ophiostoma, which is a causative agent of virulent death elm disease. So based on the carbon utilization profile, it was observed that endophytes were more efficient at utilizing whatever the carbon was applied as compared to phytopathogens. So in utilizing the carbon, they have outcompeted the pathogens. Antibiosis also, the antibiosis is the synthesis and release of molecules that kill or inhibit the growth of target phytopathogens, which results in plant disease control. The antibiotics and volatile organic compounds, they includes the ketones, alcohols, esters, etc. The volatile organic compounds having the low molecular weights, they can directly affect the growth of phytopathogens at low concentrations, although the mechanisms exactly precise mechanism we do not understand, since their perception system are unknown. Then there is parasitism also. The parasitism happens when one microbe feeds on another microbe, example a phytopathogen. So this results in a complete or partial lysis of a cellular structure. In particular, the antagonistic bacteria feed on fungal phytopathogen, cell wall material such as proteins, chitins and glucans. So the bacteria form the lipopolysaccharides and they produce cell wall lytic enzymes for the infection of their host plants. And then these cell wall lytic enzymes, they also cause hydrolysis of the cell wall of the fungal pathogen. So once the cell wall of the fungal pathogen is hydrolyzed, they are no more able to cause any disease to the host plant. So then other is the induced systemic resistance, ISR. The beneficial bacterial induce ISR in the plants, which stimulates their local and systemic defensive responses, thus protecting the host plant against pathogenic attack. Besides the classical salicylic acid based ISR, several endophytes they also activate jasmonic acid and ethylene assisted immune responses. An example of latter mechanism include the endophytic bacillus conferring systemic resistance in Arabidopsis seedling against the insect pest myzus and the green peach aphid. So here the bacillus it prevented the Arabidopsis from the insect pest. Now finally we discuss this. So the bacterial endophytes they can play an important dynamic role in the management of abiotic stresses and could efficiently be applied for environmental cleanup for sustainable agriculture development. Because the endophytes they produce several active secondary metabolites and they have positive effect on host plant growth and which ultimately resulted in the enhanced yield and productivity. Comparing with the agrochemicals, the presence of endophytes inside the host plant puts them in direct contact with the plant and they becomes more effective as compared to the rhizobacteria. And chemicals were then applied to the plant, and a large part of them do not benefit the plant and cause pollution to the environment, and some of the chemicals they mix with the soil taken by the plant, and sometimes when we consume some product of soil or the plant product, then indirectly we are consuming some part of those chemicals which are not good for our health. So, thank you so much. See you in next lecture.